month, actually, throughout New England. I go last, and I don't know what it is about the person talking about poop that's at the end. And usually I'm either right up against dinner or the end of the thing at lunch. This is a terrible idea. Why am I going? But hey, I've accepted my fate now. I'm not nearly as brave as Ellie. You could not catch me going out catching wasps. No way. I'm not as smart as her PI, Dawn Chandler. I'm a molecular biologist. I really just use pipettes and tubes to figure out the questions I'm interested in. But the questions I'm interested in answering are ecologically driven. And the stuff we'll talk about today are really pertinent to both the farm and forest industries. And it has everything to do with poop. So why might you be interested in bat guano? So if you're a bat biologist, one of the things you've tried to address, really any wildlife biologist, one of the things you try to address is what do they eat? You can usually know a lot about an animal if you just sort of ask, what do they eat? You can learn a lot about a person if you ask them, what do they eat? I don't get a chance to see what's going in. It's tricky with bats. They're night flyers, so the strategy historically has been, actually you collect their guano, but then you sit with a microscope and you sit with some tweezers, and maybe like you would in middle school, and you had the owl pellet dissection and you have to pull apart and see what's in there. You know, that's what you do, and it's painstaking work. My advisor actually was interested in doing that with birds, and so he spent 2 years of graduate school with bird poop and a microscope and tweezers, and he was able to learn a lot about what certain birds eat. The downside of that approach is obviously it's very time intensive. You need to be an expert in identifying all the different things that are coming out, and that's tricky because the quality of what's coming out isn't as good as what's going in. You follow what I'm talking about? And a lot of times, even if you are somebody who is an expert entomologist, and you've seen these bugs again and again and again, you may have just a bit of a bug, and it doesn't tell you the species of bug necessarily. It may give you something a little bit more general, like the group of organisms it's in, the genus or the family or whatever sort of taxonomic level you can get to. If you want to know whether that longhorn beetle was a Sawyer beetle that's native to New Hampshire and causes certain problems in pine trees, say, or if it was an Asian longhorn beetle and was maybe a bigger alarm, then the species level does matter, right? And so the tools that I'm going to try to do, the tools that I use, and what we'll try to talk about today, is there any advantage of going even deeper than just a tweezer could give you? And we'll use molecules, which are reasonably smaller than an entire beetle leg. So at one level, we're going to be able to look at the guano and figure out what insects are in there. And we can try to address two things at once. One thing we can address is, what are the bats eating? Sort of bigger, sort of broader question. And then the second part is, are any of those things that they are eating an invasive bug? And what ones are they? So unlike having tweezers and a microscope, and even still, if you were to do it that way, a reference collection to sort of compare what's on my tweezer and where does it fall. Just like Ellie was just showing you, you have sort of you have the beetle in front of you, and here are the dozen or so possible green shiny beetles. Which one is it, right? When I deal with molecules, I need the same thing, except I can't look at them, right? I can't look at a molecule. And the way this is going to work is no different than, say, you were going out to dinner and you sit down and you meet someone at dinner and you remember their name, but you want to call them. And then what do? Because they're gone now. What would you do? You know their name.
Look them up.
Look them up. Where would you look them up? How?
Phone book.
Phone book. This is the perfect. See, you gave two different answers. Different audiences give different ones. First audience says Facebook, Google, and the other audience says phone book. And usually somebody that says phone book, when I try to tell it to my undergrads of what I do, they're like, what's a phone book? But in principle, a phone book is a directory containing two bits of information. There's a name information and there's a phone number bit of information, right? And all you're doing is you're comparing the thing you know, the name, down a list of names, you find it, and then you go across and you say, that's my phone number, that's what I'm going to call. And maybe you're right, or maybe they're unlisted and you're unlucky. I do the same thing. I get the guano, And I'll show you a few of the steps in a second. But in principle, what I need to do is get the DNA, the molecular information out of that guano. And it's just a string of letters, which is effectively meaningless unless you have a phone book. Or you can call it a DNA book or a molecule book if you want. And in that book, there needs to be ahead of time known information associating that bug with that bug's DNA sequence. That sequence that we're choosing, and what you see up here, is a sequence that's in effectively all animals. So it's a little snippet. It's not a very, very, very long sequence relative to the size of genomes, but it's effectively a universal in animal sequence that millions of different animals have been sequenced for. And so it's a pretty big phone book. One of the books that I'm using, and there's multiple directories, is over 150,000 unique species. So it's pretty big. It's a big phone book to start with. So we're pretty likely to find things in the book. So this is how it works. In principle, you start with guano. You have different things in the guano. You can't see them, but they're in there. Perhaps there's a beetle, a moth. Obviously, the guano itself is from the bat, so there are bat cells in there as well. There's potentially the bacteria that are in the gut. All of this stuff is getting passed through into that guano pellet. And what I want to do is pull out the DNA. So I do that. But what I really want is just that one snippet that I just said, that universal barcode that is in all animals. So you'll notice it's not in the bacteria because they're not animals, but it is present in the bat, it is present in the moth, it is present in the beetle. And what I do is I make a lot of copies of that one thing so that I can read it. If I just had one copy, it might be hard to see. So in the end, I go from a tube that has guano to a tube that has just the molecule snippets that I want. And the job is to turn that molecule into something we can use in our book. And that's just DNA sequence data. So you can see the principle that we just mentioned. If you have a list of things that are in your sample, you compare them to a list of known samples, and that's the magic of the molecular biology that I do. Straightforward enough so far? Good. Sometimes it's not so perfect, though, as great of a story as this may sound. Sometimes you get situations, and I've made it with a bat here because that's something that commonly happens to me. You may have two bat species that you've sequenced, you know what their sequences are, but this little snippet we've decided that everybody has and is a useful barcode, they're really, really, really similar, such that there's only maybe two in 200 bases that are different. And you get your molecule and you're not really sure which one it belongs to. In effect, the analogy is you had somebody's phone number and you couldn't quite tell if it was a 6 or an 8. And you know that it's one of those two, but at that point you have to call both numbers. I can't say that my sequence is both bats. It is one or the other. So that can be a problem. And obviously it's not just with bats, it can be with anything. At that point you have to say, well, I don't know what the species is, but I feel pretty good that it's a bat and that it's probably one of these collections of bats. So again, as opposed to a microscopic and tweezers approach, which may only get you to typically a family level, which is still great, we're almost always getting down to the species, and when we don't, it's usually only bumped up one level. Sometimes you get sequence data that you know is absolutely true, but you have no reference for it, and that's kind of popping up more in projects when I'm getting samples back from like Nicaragua, when I'm getting samples from soils that are 1,000 years old. There's just, what was that thing? It can be tricky, but it's still a big data set. So one of the things people try to figure out using these tools, I'm certainly not the only one, is just tell me how bat diets differ based on what the two bats are. This may be useful not just if you're curious about bat biology and foraging behavior, but hey, if you're a farmer and you have a big brown bat in your farm field, and we know, using some of these approaches, that big brown bats like eating the pest in your area, that's useful. You would want to know if you had a little brown bat and it doesn't eat your pest, well, hey, that's not really helpful. Maybe we could try to attract certain bat species if there are preferences. The first thing we need to do is find out if there are. That's not that easy, though, because sometimes if you try to do this work, you have a lot of factors that are all tied together. And the question is not, how do bat diets differ by bat species, but it's of all of these characteristics that could influence what the bats are eating, really what's in the poop, What's the best predictor of what's in the poop? That's really the way to phrase the question. Is it the bat that ate it? Is it when the bat ate it? Obviously, insect communities are changing all over the course of the summer. Is it where the bat ate it? And to what degree is each of those factors important? It is a community ecologist's typical problem. It's one that I'm still struggling with, but more data is really the way to answer it, and there's no shortage of bat poop in my life. So, I'm going to simplify it for you. We're going to look at one place in Vermont. It's a place called Eli Mine in Vermont. It's a forested area, not particularly inhabited by people in any direction. And these bats were caught using big nets, and not the sort of sweeping around nets. We're talking like across the room size nets, and the bats are flying, and they fall in the net, and so you capture the bat. So you know ahead of time actually the bat species. There isn't this problem of who is it. This is a study that was conducted over the course of 1 month in one place, and I'm just going to take the data out to look at 2 of the most commonly caught bats. So it sort of simplifies all those variables since we're kind of making all things equal except for the species of bat. All right, tiny font, but I promise you I'll make it a little bit bigger. The way that this is going to work is this cluster of columns is the big brown bat. That's certainly a bat native to New Hampshire, as well as an eastern small-footed bat, one that we see as well, really sort of eastern Canada and US wide. So the columns themselves just represent the kinds of things we see. So this represents about 130 pieces of bat poop from 130 unique bats, I should say, over the course of that month in Vermont. And what you see immediately is stuff that we already knew. People have been publishing this for decades. Big browns eat beetles more than other things, and certainly more than other bats. Vice versa. Other bats in this Myotis genus, things like the little brown bat, as well as eastern small-footed, and the northern long-eared, they eat more moths than beetles. So if I was to give you a piece of bat poop, and I told you there were a lot more moths than beetles in it, you might be inclined to think it was coming from one on the right, the Myotis, than it is the big brown bat. If you have a farm stand and you have a lot of moth pests, you may not want to have big brown bats as much as some of the Myotis species. One of the other things, though, is great. So maybe this is a tool for identifying what they eat. Maybe there's potential for this to be a tool to identify invasive bugs. One of the things I think this tool would need to show is that bats have a big diet. Because if they only ate 3 kinds of moths and 4 kinds of beetles, so there's some new introduced pest, how likely is it to eat that introduced one? This is a picture that's just simply depicting every individual species that I was able to call out of those 130 pieces of poop. There's more than 130 boxes. They're flying vacuums. Like that's a bat. The size of the box represents preference, but in effect, it's really just the number of times I could find that species in a sample. And so it's still colored the same way as the previous slide. What you see is really 3 kinds of colored boxes. You see moths, you see beetles, and you see flies. We should be more specific in front of Don, dipterans. But there's a slew of these other blue shades, and that's all the other orders of insects that they're eating. And I was trying to put this up just to sort of demonstrate that in one place in Vermont for a month among 2 bat species, they're eating over 150 species that I can call. And then there's more other bits of information that I'm not calling to a species, but I still know something about its nature. So it's a broad diet. They're not like my 2-year-old daughter. Doesn't eat anything. Okay, so far so good. I really started this project with the hope that I would find them eating an Asian longhorn beetle, an emerald ash borer, or a woolly adelgid. They don't eat any of those. And I think Don was one of the first people that told me they're not going to eat those. Certainly not the emerald ash borer. And the rationale that he had is still the one that I think is absolutely spot on. Learn to listen to the adults and experts in the room. Emerald ash borer is active when, Ellie? Nighttime or daytime? Daytime. Everybody else in the room. Bats tend to eat nighttime or daytime?
Daytime.
Right, yeah, right. So sometimes, so one of this is sort of the general message is if you want to use this as a tool for monitoring invasive bugs, life histories need to sync up. And so things that are problem pests that are out in the nighttime may fall into this category. Things that are problem pests that really are active in the daytime, probably not going to work with bats. But, and I didn't put this slide in, I also work with people send me their poop from lots of different animals, including birds, and this works equally well. And although the initial approach that I took was to start with bats, now finding that we can have sort of 24 hours of the day covered if we strategically pick our organisms. So I still think there's a lot of merit to the molecular tools, but bats aren't the only way to go. But they certainly cover a sort of a broad swath of the insect community. I'll just leave it at that. Are they eating any pests at all? Yes, they're definitely eating different pests, but if you look at the relative abundance in which I detect these types of pests over all the things that bats are eating, they tend to be fairly small. And again, if you think of these bats as flying vacuums, what I see in their guano again and again and again are the kinds of insects that come out and bloom. So the top hit was a fish fly, the second top hit was a mayfly. We see these in between 30 to 15% of all samples. And these are the kinds of things that emerge in like, you can track them with NexRad radar, right? That there's just enormous amounts for 2 or 3 days, and that's what you see in the guano. It doesn't mean that bats necessarily are preferring that one species. That just means that's what the bat ate that weekend. They're obviously eating many, many, many other things. So as an invasive detection tool, I think it's not going to be the most effective at finding very rare things. I think what it's going to find are the things that are abundant. But that doesn't mean it's not a useful tool. And so what you can see in the second slide here is we're picking up some pine beetles that just aren't found here. Kyle Lombard, who's talking 2 rooms over, or was talking 2 rooms over when I asked him about that specific one, because it popped up in Fox State Forest among other places here, mentioned that there really isn't much record of it. But it is a pest, and we should know about it. And so even if this isn't necessarily finding the things you absolutely were worried about, it is a tool that will let you be aware of problems that you may not even know exist. And that's a different approach than how we monitor pests now, right? Right now we set out pheromone traps that we know exist. We do aerial surveys for things we know could be a problem. But this is a tool that's relatively agnostic because the bats have such a big diet. So I think it offers some useful, it certainly is a useful tool. So just to recap, whether you're talking about a forested or an agricultural landscape, if you have bats in your area, it's very likely that they're going to be eating the things that are in abundance. It is also likely that they have the potential to eat the pests that could be in your area. I have an agricultural study going on in partnership with Rutgers University, where all the guano is now processed and about to be sequenced. We had 11 farms over the course from April to October, and we're going to try to see, can we predict effectively what the pest would be in real time. And so we have that year-long data, and the idea now is, hey, if you know what your pest is, and you can do this whole thing from start to finish in 1 week, you can then set out the appropriate traps and try to get rid of the problem before it's too late. So happy to take more questions. Thank you to, the cool thing about this is that, I didn't mention this, I didn't do the collection. Virtually probably like 10% of the collections, 90% of it was volunteers. There were people that already had bats in their barns that wanted to get rid of the poop. And I said, oh, I'll take it. So this is largely a citizen scientist venture. And it's one that I think it's scalable, right? It's hard as a graduate student to get out and do more than 2 sites because field work is labor intensive. But if you can leverage the fact that people already have these kinds of I don't know how many people are going to want to scoop up bird poop, but still, it's something that you can have a massive survey. And there's bats everywhere in North America, let alone the world. So it's something that can be broadly applied. Same molecular tools will work equally well. Thanks, guys.
Thank you. So I'm sure Devin would take a couple of questions, but as I know some of you will be moving on, I just want to thank all of our speakers this afternoon, because this is one of my fun times of the year. I get to listen to the people who are actually doing the research, and they have wonderful stories to tell. So we'll take questions.
Sure.
So Devin, so you're talking about farms. So are you also doing any light trapping to see if insects that might be night flying, so what you're getting from the bats might actually be being collected from the farms, or are they being collected elsewhere than right over the farm?
The proper way to design this study would have been to collect both the bat guano and as much of a representative sample of just what's in the area and compare, are they eating what's available or do they have a preference? And went over to talk to Don about like, how do I catch insects? The problem was manyfold, but principally, I didn't do it. So that's the first thing to mention. No, I didn't do it because we felt like if we were catching the flying insects, that would bias our comparison, because I'm finding bats eat a ton of ground beetles, which don't fly too much. So I would need something which exists that would capture the ground and the flyers. And if I have them attracted to the light, maybe they're ones that are, no matter what sampling technique I use or even a collection, I was never going to get a truly representative one. I also have 20 different sites that I'm doing this at, and there was just no way to ask the volunteers to do it. I think in retrospect, I should have just made a simpler study and just done one site and made that effort. But now it was more of, let's just see what's out there and see what's in there, I guess, really. It's not a representation of what's out there. You are biasing to what the bats will eat, but it's so broad that I felt like I'm not too worried now. Is that reasonable? Okay, great. Yeah.
The study that you're doing with Rutgers, are some of those farms in New Hampshire by any chance?
They're all Pennsylvania, New Jersey for the first round, and as the grant was written, it's supposed to now expand into New Hampshire either at year 2 or year 3. Yeah. It's paying my bills, so I'm happy to work with as much New Hampshire as possible.
So the caterpillars, like tent caterpillars, so are they actually eating the larvae or are they eating the adults?
Yeah, how would you have to figure that one out? Just know something about when the insect emerges. If we can find them in April, then they're probably eating the larvae. If we can find them only in August, then they're probably eating just the adults. So 2015 that I showed you, that was the data that I mentioned. I didn't start collections until June because I didn't start in the lab until June. But 2016's data, which is actually getting sequenced today, that will answer the question because we started in April. So I wanted to address that, and every time I've given this talk, I've had to add this caveat, and I'm so excited to give another talk later that will finally answer that question. Yeah. Great. Thanks, guys.
Thank you.